Hello, welcome to our presentation. My name is Chairinda Dachwan. I am one of the members of the researchers team from University of Indonesia. Our study title is Comparison of Real-Time BioCov-19 RT-PCR Kit with N1 and 2 CDC RT-PCR for detecting SARS-CoV-2. As we know, in December 2019, several cases of pneumonia of unknown etiology have been reported in Wuhan, Hubei province, China. And then we know the etiology is SARS-CoV-2, and the WHO announced that the disease caused by the SARS-CoV-2 is referred to as coronavirus disease 2019 or COVID-19. Based on the WHO and the CDC, an examination using the molecular test method is the recommended examination for diagnosing COVID-19. One of the methods used is quantitative real-time PCR, and currently there are many kits for detecting COVID-19 that are recommended. One of them is BioCov-19 RT-PCR kit, which is a real-time RT-PCR reagent that detects the COVID-19 gene in samples like oropharynx and nasopharyngeal specimens from patients with respiratory tract infection. Using a one-step real-time PCR system and Techman probe to detect COVID-19, RDRP gene and N2 gene and RPP30 gene as internal control and extraction control. Many COVID-19 RT-PCR kits are currently commercially available. An independent assessment of this product is not yet publicly available and barely needed to guide implementation of accurate tests. One of them is BioCov-19 RT-PCR kit that produced in Indonesia. So the aim for this study was to compare basic analytical and clinical performance of BioCov-19 RT-PCR kit with N1 and 2 CDC RT-PCR as the standard for diagnosing SARS-CoV-2. For the literature review, SARS-CoV-2 has been classified as a beta coronavirus of group 2B with higher similarity, over 96% identical at the whole genome level to two bat-derived coronavirus strains than to other known human coronaviruses. SARS-CoV-2 is an enveloped, non-segmented, positive sense RNA virus that is included in the cervical virus orthocoronavirinae subfamily, which is broadly distributed in humans and other mammals. Its diameter is about 65 to 125 nanometer, containing single strands of RNA and provided with crown-like spikes on the outer surface. Rapid and accurate COVID-19 detection is critical to controlling outbreaks in hospitals and communities, and according to the current diagnostic criteria established by China's National Health Commission and WHO, laboratory examinations, including nasopharyngeal and oropharyngeal swab RT-PCR tests, have become the standard assessment for the diagnosis of COVID-19 infection. To identify patients earlier, two one-step quantitative RT-PCR assays were developed to detect two different regions, such as RAF1B and N of the SARS-CoV-2 genome. For the methodology, we are doing limit of detection tests, clinical sensitivity and specificity tests, and cross-reaction tests. The minimal detection test used standard genus SARS-CoV-2 RNA vs ampliran SARS-CoV-2 RNA control containing first cell SARS-CoV-2 RNA that consisting of 14,000 copies, and to determine the minimum detection of the BioCov-19 RT-PCR kit, dilution of first cell SARS-CoV-2 RNA was carried out using 1/10 and 5/10 buffers from ampliran SARS-CoV-2 RNA control, respectively. Dilution of your cell SARS-CoV-2 RNA, 14,000 copies into 1,400 copies, 140 copies, 14 copies, 7 copies, and 3.5 copies. And each dilution was tested for real-time PCR using BioCov-19 RT-PCR kit And the lowest dilution result that can still detect SARS-CoV-2 RNA is the minimum detection value from the BioCov-19 RT-PCR kit. A panel of clinical samples with in-house confirmed SARS-CoV-2 between 15.05 to 38.50 cycle threshold for the N1 and 2 gene during initial diagnostic were prepared, and the number of samples is 135 samples. From the comparison between the real-time BioCov-19 RT-PCR kit with N1 and 2 CDC as a standard, the sensitivity, specificity of the BioCov-19 RT-PCR kit will be obtained. And to determine the potential cross-reaction of the BioCov-19 RT-PCR kit against microbes that have the potential to cause false positives, a cross-reaction test was carried out against 12 bacteria and 7 parasites and 3 fungi. For the result and the discussion, for the limit of detection result of the BioCov-19 RT-PCR kit was within the 5-fold range dilution, which was 7 copies per milliliter. And for clinical performance of BioCov-19 RT-PCR kit was determined by testing 80 positive and 55 negative specimens for SARS-CoV-2 virus, and the result shows sensitivity of 97.5% and specificity of 98%. The comparison of BioCov-19 RT-PCR kit with N1 and 2 RT-PCR shows in the table. BioCov-19 RT-PCR kit does not cross with the common respiratory pathogens that mentioned before. So from the conclusion, here we provide a comparison of BioCov-19 RT-PCR kit for the detection of SARS-CoV-2 in clinical samples. BioCov-19 RT-PCR kit has good sensitivity and specificity, which is 97.5% and 98%, has limit of detection within 5-fold range for 7 copies per milli, and does not have cross-reactions against other respiratory pathogenic microbes. So it can be used as a choice of a real-time RT-PCR reagents for diagnosing SARS-CoV-2 in community. We acknowledgement for the Ministry of Research and Technology of Indonesia, or RISTECPRIN, for funding this research. This is the end of our presentation. Thank you for your attention.